Welcome, ladies and gentlemen. So today we are going to start chapter 16, which really is going to go over the basics of what DNA is and how your cells actually go about making copies of that DNA. So when we talk about DNA, right, this is the substance of molecular inheritance. This is the hereditary information. It's the set of instructions for your cells on how to look, how to function, and it's what we pass from one generation to the next. So all of this information is coded in the chemical language of DNA. What's the sequence of the A's, T's, C's, and G's? And that code then gets translated into proteins, and those proteins are what drives your cell's structure and function. We need to be able to make copies of this DNA in order to make more cells as well as to make more organisms. So this directly implicates the development, the differences in traits we see from one cell to the next in an organism based on how the cell is expressing that information, as well as the differences that we see in different individuals in the same species, as well as the differences between species. But it's a universal language. So every organism on Earth, at least to our knowledge, is based on DNA. And then it's just a question of what's the sequence, how much do they have, and how do they use it. So before we get into the rest of that, let's just take a step back and figure out how we got here. Because we didn't always know that DNA was a thing, let alone what it did. And in fact, it's only been within the last 100 years or so that we have actually figured this out. So there's a series of what we kind of call classic experiments in molecular biology that really sort of give us the picture as to the building blocks we worked on to understand what DNA was, what it did, and then how did it ultimately actually work in the cells to drive its structure and function, as well as how did we copy it. So we're going to start with this first one back in 1928 by a gentleman named Frederick Griffith, and he was working with Streptococcus pneumonia. So this is the bacteria that is known to be responsible for bacterial pneumonia. All right. Um, Not to be confused with viral pneumonia, by the way. So when we look at this, there's basically two strains he was working with. Your smooth strain, which has, if you go back to your microbiology, right, these are prokaryotes. So they have a cell membrane, a cell wall, but then they can also have a capsule, sort of extra sort of carbohydrate layer on the outside of their cell wall. And that capsule can do a lot of things for them, one of which can be to help evade the immune system of their host. So this smooth bacteria has this extra layer, has this capsule, and as a result gets around the organism's immune system, and so it's pathogenic. All right. So what Griffith did in working with those smooth bacteria is he created a control. He injected the mouse with that smooth strain bacteria, and it did exactly what he anticipated. It killed the mouse. He also took it one step further. We were at a point in this stage of history where we were starting to understand germ theory and how to keep people healthy and what those germs were and how to prevent you from getting sick from them. And one of the things we had figured out by this point was that if we heated things up most of the time, that that stuff wouldn't kill us. So he heat kills, he puts this bacteria into a test tube, into a petri dish, however he accomplished this, and warmed it up, right, to pretty significant temperatures. And so he heat killed the bacteria. So he injects that into the mouse and it does exactly what he expects. It was successful in killing the bacteria. Remember, things like enzymes are susceptible to heat. So if we heat it up, we destroy the enzymes, we hopefully destroy the ability for the bacteria to continue reproducing, and the reproduction is really what makes it pathogenic. Okay, we go to the other strain he worked with. He was working with this rough strain. This rough strain lacked that capsule. So it had a cell membrane, had a cell wall, but didn't have that additional layer As a result, the host's immune system is able to take care of it, and it's generally harmless in most healthy hosts. So he does one more control, inoculates that mouse with a living R strain, and the mouse's immune system does what it's supposed to, kills off the bacteria, and survives. The final step of his experiment is where things got a little weird. This was not expected. So he took a mixture of heat-killed S strain, right? Remember that allowed the mouse to live, right? That heat killed off that bacteria, didn't destroy the mouse. And he mixed it with live R strain. Also, gave that to the mouse by itself, mouse lived. Put them together, and he ended up with a dead mouse. What? Right? So how did two things separately not kill a mouse, but collectively did? And what Griffith didn't understand at this point in time is how that process of transformation works, right? So if you remember bacterial transformation from earlier this year, bacteria are able to take genetic information out of their environment, incorporate it into their own genome, and express it. So remember that everything about a cell, both structure and function, is genetically controlled. So that capsule would be something that would be genetically controlled. So Griffith recognized some sort of transformation was happening. Something was happening in that test tube that was allowing these cells to now become pathogenic. And what we will eventually learn is that transformation process. So what he didn't understand is by heat killing that bacteria, you had to heat kill it enough to not only destroy the proteins, but if you don't want the transformation to happen, you also have to heat it up high enough to destroy the DNA. That's not what he did. So those bacteria were able to take up DNA from their environment, incorporate it, now produce that capsule, and now become that S strain. But again, here in Griffith's point, all he knows is something happens. He calls it transformation, but he doesn't understand at this point what's causing that transformation to happen. That's going to take some time. All right, so the next set of experiments isn't going to come until 1944, and it's going to happen with a gentleman named Avery as well as a few other colleagues who are working with him. And Avery sort of rediscovers Griffith's experiment. He's directly observing that transformation now between that R and S bacteria. And he's looking at this transformation that's happening, right? So he performs a series of tests to determine the material. Is it DNA or protein? By the time we get to 1944, there's enough consensus that genetic information that is passed from parent to offspring that connects their characteristics together had to be one of these two macromolecules, right? So DNA or protein. And so Avery's running all sorts of tests, qualitative tests showing it's DNA, chemical analyses, proportions of elements showing that it's DNA, enzyme tests showing that it's DNA. He announces to the world that this transforming material is DNA, and most people laugh at him. And just think of the context of that. So we at this point in history understand that genetic information, whatever it is, has to be able to be responsible for all of the diversity that we see across every organism we've ever come across. Even if you take into consideration the sheer diversity in traits we see within our own species, that's huge. And now we're going to expand that out to every organism we've ever come across, and we're starting to understand viruses and bacteria and things that we can't even see with our own eyes. Holy crap, there has to be a lot of variation in this code to make all of that variation in these traits. And so when you boil that down to that understanding, think about the number of building blocks we have in DNA versus the number of building blocks we have in proteins, right? Just think about that. So when we talk building blocks for DNA, right, we're talking nucleotides. Okay, so we're talking nucleotides here. Well, how many different nucleotides are there? Well, if I just talk DNA nucleotides, then I'm talking 4, right? Your A, T, C, and G, right? Those 4 nitrogenous bases. I go to my proteins, right, and their building blocks are amino acids. Well, think back to your biochemistry. How many different amino acids are there? 20 of them. So just think about them like letters in an alphabet. If you had 4 letters in an alphabet, how many words could you make compared to 20 letters in the alphabet? Well, of course they assumed that proteins must have been the more likely candidate because you had so much more options to choose from. What they didn't understand was how the code worked. So we're going to get a little bit more credence, a little bit more support for Avery's findings when we look at things in the 1940s and 1950s. So Shafgrad, he's going to be the one who ultimately gives us Shafgrad's rules, or our base pairing rules. So he's just off studying DNA composition. He's understanding that it varies from one species to the next, but what he is seeing is that there is these 4 different bases, and those bases always come in very specific proportions, right? So I'll always see the same number of A's to T's. I'll always see the number of G's and C's identical. And so he's starting to figure out how this genetic code might work. Doesn't have all the answers, just understands proportions of molecules, and is starting to give some more credibility to the potential that DNA is our genetic material. By the 1950s, we know that DNA is a polymer of nucleotides. We know that each nucleotide consists of that nitrogenous base, a sugar, and a phosphate. Add that to what we now know from Shafgrad and the proportions of those bases, they start to give us some more information. It's going to take until 1953 before an experiment finally comes on the scene that shuts the door on this, confirms that DNA is in fact the transforming material. And so this is going to be performed by 2 gentlemen, Hershey and Chase, and they are working with these T2 bacteriophages. They're viruses that infect bacteria, right? And the cool thing about viruses and why it was such an elegant choice, simplistic choice for this kind of experiment was that viruses are only made of 2 molecules, DNA and protein, especially when we look at bacteriophages, right? We get into animal viruses and then there's some extra things going on in their lipid coats and things like that. But if we're talking bacteriophages, nothing else. It's DNA and protein, period. All right, well, those are the 2 molecules that we know are potential candidates for this genetic material. We also have a basic understanding of how viruses infect bacteria and how they reproduce, right? So we look at viruses, we know that they inject information, instructions into their host cell. That host cell then makes copies of that virus, and that virus then bursts out of that cell. That's how the infection pathway works. We know the basics of that by this point in history. So Hershey and Chase set up an experiment in 2 parts. Their first experiment, they label with radioactive sulfur the protein coat, okay? So remember that we can find sulfur in the R groups of an amino acid, right? So if I look at the basic structure of an amino acid, right, it looks like this. There's your carbon, your carboxyl group, your R group, your amino group, and then that hydrogen, right? So that sulfur can be here. I look at the basic structure of a nucleotide, right? My phosphate group, my pentose sugar, and my nitrogenous base, all we end up seeing, oh, sorry, that drawing isn't awesome. All we end up seeing in these guys are the phosphorus, the nitrogen, the carbon, the hydrogen, the oxygen, right? So there's no sulfur here. So that means that they can use that radioactive sulfur to know that it's only going to attach to the proteins because they do know the basic chemistry of these molecules at this point. It's going to stick to the protein, not the DNA. Okay. So they label the protein, and then they just basically let the virus do what it's going to do. They put it into a culture of bacteria. The viruses do exactly what they're supposed to do. They infect the bacteria, off they go. They examine the cultures then. They basically blend it up, centrifuge it, and separate out the material. So when you do centrifugation, right, you basically stick a test tube into a centrifuge that spins it really, really fast. Everything separates out by density. So all the heavy stuff like the bacteria and the cell components are going to sit down at the bottom. All of the sort of viral parts that aren't directly connected to the bacterial cell itself will float to the top. So when they looked at the centrifuged test tube, they looked for where the radioactive material was. Was it down in the bottom of the test tube, which we would call its pellet, all the heavy stuff, which is the cellular components, or was it up in what we call the supernatant, so more of the liquidy stuff, the lighter stuff? And what they found is that there was no significant radioactive material in the pellet where the bacterial material was. It was all up in the supernatant where the coat of that protein, or excuse me, that coat of the virus should be. So by this point, they've pretty much concluded that the infection material is not protein. Could have stopped here, but went one step further just to ensure that they understood how this worked. And so they did another experiment, this time labeling the DNA with radioactive phosphorus. So go back to my drawing here, right? There's phosphorus here in this group here, but there's no phosphorus to be found in an amino acid. So we know it's only going to stick to the DNA, and we run the experiment again. This time, when they centrifuged everything out, all of the radioactive material was down in the pellet where that bacterial material was, not up in the supernatant where all the additional viral lighter components would have been. And so this gave us conclusive evidence that the information that those viruses were sticking into the cell was the information necessary in order to make more viruses, and that information was, in fact, DNA. So this picture just kind of does a nice job putting all of that together, right? So here's that first batch. Sulfur attached to the proteins. We allow them to infect, blend them up, centrifuge them. All of the radioactive material is here in the liquid, which is the protein components, not down here in the pellet, which is where the bacterial components would be. Second experiment, just to confirm that their understanding is correct, we radioactively label the phosphorus, which sticks to the DNA, run the experiment again. This time, all of that radioactive material is down in the pellet with the cells, not with the rest of that leftover viral component. Boom. DNA is our transforming material. This is what's responsible for genetic inheritance. So we now understand by 1952, that DNA is in fact the genetic material, but we still have no idea exactly what it looks like, how it works, or how we replicate it. And so that's going to take some information from these scientists here. So here's Rosalind Franklin. All right, and she is actually going to create this picture right here in 1953, something that'll ultimately become known as Photo 51. And she is an X-ray crystallographer. So basically, she'll stick genetic material or any other material that she wants to find the structure of into crystals and shoot X-rays through it. And what comes out are pictures like this. Don't ask me exactly how it works. Don't ask me exactly how to interpret it. Not my thing. But essentially what they were seeing here in terms of the measurements she could take from one band to the next across these bands here ultimately was the information necessary for these two gentlemen, Watson and Crick, and her interpretations of this picture creating it to understand that DNA had to be a ladder. And not only did it need to be a ladder, but it was a parallel ladder and that it was uniform. All right. So Watson and Crick are working in and around the 50s to try and figure out what the structure of DNA looks like. And they are putting double-ringed bases together. They are putting bases on the outside. They are putting single rings together. They have no idea how to make this work, right? Nothing seems quite right until they get a hold of that picture. And what they end up realizing is that it has to be this parallel ladder, which means that I have to have a double ringed base connected to a single ring base every single time. So I only ever have 3 rings of nitrogenous base across the span of the interior of that ladder. And that's how it's going to be nice and parallel and even all the way through. So that means that A can only pair with T, C can only pair with G in order to meet that 3 base across, as well as be consistent with Scharfgrave's proportions that he was finding back in the 1940s, sticking with those base pairing ideas. Here's the interesting component to this story, however. Those 2 gentlemen didn't ask Rosalind Franklin for that data, didn't give her credit for that data. All right, she was a woman working very much in a man's field. As a result, was certainly not willing to take people's crap, was not necessarily willing to share her information either. She fully understood that anybody who utilized information got credit for it. Whoever figured it out was the one who got the credit. And so she was very protective of her work. Wasn't necessarily one to just go, hey, here's my information and share it with the world. And so what ultimately ends up happening is Watson and Crick do find out that she is in fact working on this information. She is not the head of her own lab, and so they get a little sneaky and end up getting this data from her lab manager without her knowing, develop the model of DNA that we now know, and never give her credit for it. She is not mentioned in the Nobel Prize that Watson and Crick end up winning, at least initially, for this. And it doesn't come out until after her death in a book that I believe it was Watson ended up writing called The Double Helix, where he tells this whole story of how they figure this out. Ends up naming her in this is pretty much point blank, like, yeah, we stole the data, like, whatever, like, who cares? Not particularly apologetic. If you hear him talk about her, he is not particularly kind to the way he paints her. There were a lot of people who came out after the book was initially written being like, that's an unfair characterization of her. Regardless, she didn't get credit initially. Now there's some more credibility to the credit she should have gotten. Whether or not we can ever sort of right the past is another question. She definitely ended up dying, in fact, of cancer, probably likely from all the radioactive material she encountered in her life, and died without even knowing that she was a major player in the discovery of DNA. So kind of just an interesting story, depending on what side of things you want to come down on.